So I thought, I kind of racked my brain for a while, and I thought, what am I going to talk about? And then I realized I have a fairly recent NSF instrumentation award to build an instrument that has just arrived at the University of Colorado, or the second half of it, and that's what I'm going to talk about today. So really the goal of my work is actually kind of similar, but I take different approaches to what Michelle was talking about earlier, which is overcoming some of these challenges that we have in nanoindentation or small-scale mechanical testing of biological tissues and biomaterials, because they're complicated. Like George said earlier, they don't have great surfaces. Sometimes we find that there's thin films of fluids at the top, but principally, they're very complicated for several reasons, but they're also interesting because of these reasons. So as Michelle can talk, I think, even better about, you know, these materials are hyperelastic, viscoelastic, poroelastic. They have all of these really complex properties that if we can understand those properties, they can help us develop really appropriate constitutive relationships that we can then use as inputs for models. But getting those at these length scales are really complicated, in part because within these biological materials, there are functional gradients, meaning that we have one set of properties in one place. Right next to that, we might have a graded set of properties, or maybe even a complete different set of properties. They're hierarchical, which means that they have a large scale structure, a mesoscale structure, a microscale structure, a nanometer scale structure, and at each of these different length scales, there are different properties and different constitutive relationships, and I think that was illustrated by Michelle really well earlier because of the porosity effects, right? Most of these materials are not homogeneous, they're composites, so they have different constituents. So you just think about bone, where it has a water phase, which people tend to ignore. It has a organic phase, which is primarily type 1 collagen, and it has mineral. That's a composite material by my definition, and so it has composite material properties, and so needs to be considered as a composite material, as opposed to a homogeneous material, which the very useful formulations that we use for nanoindentation, all the Oliver Farr, et cetera, assume homogeneous material composition. And because of this heterogeneity, it gives us the ability to hopefully extract some information about the different constituent phases, but also it's not just heterogeneous from the composite standpoint, it's also heterogeneous from the standpoint that it's almost like a material that has different phases. So like a steel, where we have grains and grain boundaries and things like that. So we have different structures, and within each of the structures, we often have different material properties, different microstructural compositions. So we might have fibers aligned in lamella that point in different directions, and factors like that that we need to take into consideration. So in other words, we can't just assume that because we get a really nice flat material half space when we prepare a bone sample, for example, which is the biggest source of study in my lab, we have to assume that there's a lot else going on. So the question really is, how do we study that? And how do we partner other methods with nanoindentation to learn more about these materials? I just want to show you some examples of materials that we've studied and the complexity here. This is actually a PEG dimethacrylate hydrogel, where we've been working with Stephanie Bryant in our chemical engineering and biological engineering department at CU Boulder to make osteochondral or bone cartilage types of materials. I'm also working with Brendan Harley on this, where we have a stiff, highly cross-linked bone-like side, a cartilage-like side. We're now putting fibers into these things. We're doing micrometer length scale 3D printing of structural elements to try and stiffen up the hydrogel construct, which means that we're now basically making composite hydrogels, and they're crazy complicated. We're trying to figure out how to appropriately test them. We've also studied bone, and I'll show you just a little peek of this data later, but this is just showing an osteon in cortical bone. You can see that in the middle there's a big hole. That's one of the Herversian canals or fluid-filled canals Michelle was talking about earlier. And there's also other teeny tiny little lacuna or lacunar spaces in there. But you can see the indenting over this region with just a spherical tip and multiple indent depths. We're sampling different volumes of the material, which means we're sampling different structures. And we're moving across these hierarchical length scales. And just to further illustrate the hierarchy in tissues, this is an acid-etched region of bone from an SEM image my postdoctoral advisor, Ellen Boyd, took in London, where if you remove the bone mineral, you can see the underlying collagen matrix, and you see these branching collagen fibrils, which also show the porosity that Michelle was talking about earlier. But if you see the 5 micron scale bar down there, depending on my indent parameters, if I go to different contact depths, which means I'm sampling over different contact areas of my tip, look at how much different material I'm sampling. I could be sampling the fibers themselves. I could be sampling small pore spaces like you see in that middle circle, or I could be sampling very large pore spaces. And so the indent response, and I'm so glad Michelle gave her talk earlier because it sets this up perfectly, the indent response is very dependent on the parameters that we put in. And so we can interpret a property as a bone property or a hydrogel property, but really it's a bone property for the specific set of parameters that we used. It's not necessarily a material property. So we need to really figure out how the indentation parameters tie into these things. What I'm going to talk about today is also how the structural and especially the chemical or the material parameters tie in as well. Okay, so I'm going to focus on this heterogeneity, where it comes from, and what we do about it. Okay, so just taking a quick look at human cortical bone. This is a nice study that came out of Christine Ortiz's lab at MIT a couple years ago, where they looked at both transverse sections. So this is if you take a bone, like a femur in your leg, and you cut perpendicular to the long axis of the bone versus longitudinal along the long axis, you can see that structurally they're different. In the transverse view, you're looking down the holes that Michelle was showing earlier, and longitudinal, you're looking along the holes. Turns out the material is, of course, aligned in different ways in these two planes. And so we have this heterogeneous structure that really changes what the behavior of the bone looks like in the two different planes. We also know that the heterogeneity plays a role in bone, as it does in many tissues. It turns out that this paper really nicely showed by mapping over these 2 by 2 micron regions using AFM-based nanoindentation, and then comparing their AFM-based nanoindentation. Do I have a pointer up here? If I don't, that's okay. Oh, you've got one? Okay.
It goes intermittent on it.
That's okay. I'm up for the challenge.
When it goes intermittent, just unscrew it a little bit.
Disassemble, rebuild. So these black spots here are actually from the AFM-based nanoindentation. Everything else is basically from using Berkovich tips on a conventional nanoindenter to assess the bone. And what you see here is a lot of noise. And basically what happens is that as they penetrate to greater indent depths, they're seeing a decrease in the coefficient of variance. That's because what you're really doing is you're averaging your properties over a larger area, so you get away from some of this heterogeneity. So in other words, this is where you see a high coefficient of variance, so you see a high heterogeneity, and that decreases as you go to greater indent depths. So in other words, greater indent depths, you're losing sensitivity to that heterogeneity. What they showed in addition to this, because they also did some fracture toughness testing, which isn't really the purpose of this talk, but why is the heterogeneity there? Well, in bone, it's because it probably maximizes energy dissipation, where you get a high stress state in advance of a propagating crack. So in other words, heterogeneity stops crack propagation, it stops damage in bone. So that's really important. But the problem is that when I just go in and do nanoindent tests, as I'll show you here on this next slide, without any kind of additional analyses, what I see here, if I take their transverse section and longitudinal section, and then a transverse and longitudinal section from my own research with Alan Boyd and Andy Bushby in London, is that if I look at any one of these regions, I see this is a histogram of elastic modulus over an indent region where I use the same tip, the same indentation parameters, et cetera. And you can see a wide spread in this data. I don't get one value for elastic modulus. And of course, we would expect a histogram, some sort of Gaussian-like distribution, or maybe skewed distribution, because it's biology, you always get that. But the problem is I don't know what that source of heterogeneity is. So what we did in my postdoctoral research is we took a stab at this, and we said, well, in bone, it has varying degrees of mineral volume fraction. So we use this quantitative backscatter electron imaging technique, where using standard materials that we image every time we image the bones, we can basically threshold the images to all be the same. In other words, we can correlate the amount of brightness in these images here and here to the degree of mineral volume fraction. So the take-home message here is if it's black, it has 0 mineral volume fraction. If it's white, it has 100%. So everything here is kind of in the middle, except for this is where cells and marrow space is. So of course there's no mineral. In here you can see this is pretty bright, so that's got higher mineral, less, so on and so forth. So you can see bone adjacent to these canals is more newly formed, it's younger, has less mineral. The existing bone here in these interstitial spaces has more mineral because it's been around for longer. It's a thermodynamically driven process over time. We'd expect that to happen. And it turns out that we do see correlations that I'll show you in a minute. But this is one instance where you think, okay, what other techniques can we use to understand this? Because principally, quantitative backscatter is really nice. It tells us how much mineral is there. But if you know anything about bone and mineralization, you would say, but wait a minute, what kind of mineral is that? Is it octocalcium phosphate? Is it hydroxyapatite? So the mineralization people, they have a whole list of minerals that they go through. And I always say, I don't know, it's just mineral, it's bone mineral. Isn't it the same? And then people get really all up in arms. So you can use techniques such as Raman spectroscopy. This is one of my favorite bone Raman papers, because I'm a little bit of a geek like that. But my friend Eve Donnelly, who I spent the weekend with at the Orthopedic Research Society conference, she did this paper as part of her PhD, where she went into these rat bones, and they have this region of bone here on the edge that she's showing in this blow-up section, where she went in and did nanoindentation. And these colored lines are actually real. What you do is while the rat is alive, you can administer tetracycline or other fluorescent labels that they're chelating agents, so they deposit into the bone as the bone mineral is being formed. So in other words, on the day that this red label was given, it deposited into the bone for the next 6 or so hours. The same with this yellow label, orange label, et cetera. So she can know exactly how many days, so these are numbers of days, 0 to 4 days old bone, 5 to 14, et cetera. And so she can go and indent in different regions of different ages of bone. Okay, and so by doing this, what she can do is she can map out the properties. So she was also working with a wonderful woman named Adele Bosky. Adele is an FTIR and spectroscopy expert in New York. And so Adele basically helped to make these thin sections. So FTIR requires thin sections. They did some FTIR. They also did Raman, where you can use the bulk sample. And I'll talk a little bit about that more later. And so they can plot out, or they can measure using spectroscopy methods, how much mineral and how much matrix, or the collagen and other organic molecules are there. And you get these peak ratios. And so you can report the results of the peak ratios. In other words, you can get an idea of why the properties in this region are different than the properties in this region. And it turns out these have a very low modulus, these have a higher modulus, and you can correlate that with the chemistry. This is the calcium to phosphate ratio, because bone is calcium phosphate. It tells you a little bit about the crystallinity and the aging of the bone. And so indeed, when we look at some of her results, we see indentation modulus here on the left. If you look at the mineral to matrix ratio, as you increase the amount of mineral, you increase the modulus. It's a composite material. We would expect that to happen. And as you change the carbonate phosphate ratio, I won't go into this here, but I'm happy to talk about it later, you also see a correlation. So in other words, using tools like quantitative backscatter and Raman spectroscopy, we could start to dig into the underlying reasons why we see changes in these highly varying properties in biological tissues. So not just bone. So let me show you one more example of why we need this type of thing, and then I'll go into tell you what we're doing about it. Okay, so this is a study that a couple of my students did a couple years ago, where we went and we took a range of tips. We started with a Burke, and we looked at a 5 micron, 25 micron, et cetera, all the way up to a huge 200 micron spherical indenter tip. So we used Burke and conicospherical indenters. And we basically tried to understand, when we were indenting into lamellar bone, or that layered bone that you've seen in a couple of these images so far today, what is the influence of the indenter tip shape and size in indenting into these samples? Because of course here, as Michelle nicely explained, you get some plastic deformation, plastic-ish deformation, you get elastic deformation and some viscous dissipation, and you also get mostly elastic with these tips. But you can also see that as you go into these greater depths, just looking at some spherical tip and the Burke, or maybe a very small radius spherical tip, I might be indenting within one lamella, or I might be indenting over others. So I threw these slides in this morning because I thought this would be really relevant to what we're talking about today. So I'm not so prepared to talk about it. But the point is that as you increase your indent depth and you also increase your indenter tip size, you basically lose sensitivity to this heterogeneity. But the question still remains, because I can look at the data from these tests and I see, as I see lots of heterogeneity, less heterogeneity. So this is kind of like what we were seeing earlier in my talk. But the point is we don't have any context as to why these properties are changing. And so what happens in a lot of bone papers in the literature, and I think that this was fine early on, but now in this day and age, in some ways there's not really an excuse for it, of collecting a bunch of indentation data, but not explaining why the modulus is changing. And so that's really a common phenomenon. So we're trying to tackle that issue, yeah.
Is there a theory that you had in the last slide, you showed like a bigger, bigger tip will encounter more of those vertical strips?
Right, so why is it getting stiffer? Is that what you're wondering? Or
is that taken into account in any theory?
Is it taken into account in any theory as to? Yeah, you know, maybe we should talk after because we wrote a whole paper on this. And we use some finite element modeling to explain that, that phenomenon. So let's talk later just so that, is that okay? Okay, thanks. So yeah, so we, you know, having come from this world of combined measurements where I ran between a nanoindenter and a quantitative backscatter, well, an SEM, and then talking to Eve this weekend, and she was telling me how difficult it was to collect that data and find the indent locations again on her Raman setup. We could see that this is really useful, but it's hard. So we've used this, as I showed before, of collecting quantitative backscatter electron images. This is a human femoral head. And what we do, the process is this, is typically you take a basic image like this, where I get a survey, and I decide I want to indent, say, over here or over here. I'm talking centimeters at this point. This human femoral head, I don't know, is like 3.5 centimeters wide. So now I take that over to my nanoindenter where I maybe have a 20x objective, and I try to find a region that I found on my SEM. And so I find a suitable region like this. I put some indent arrays, which these are big marker indents. My real indents are in the middle. I've plotted that out over here over the quantitative backscatter electron image. And then I have to come up with some MATLAB code, or we used to use more crude methods in the old days to do this, and I can correlate the spots. So here this is just showing, again, my nanoindents at higher mag, and then where they lie on my regions here. So from this region, again, I can get a mean gray level, or we now correlate it to a mineral volume fraction. That's very useful because we can put together plots like this, this is work Michelle and I did years ago, where we looked at a whole range of samples of different mineral volume fraction, and Michelle had the clever idea of bringing in the composite bounds notion to try and explain why the data was doing what it's doing. But we still don't really know, aside of mineral volume fraction, why these measures are changing. So what we've got here is basically a really nice system that takes a lot of work to achieve the end goal, even with newer MATLAB programs and things that my students have been writing. So I do want to say that for quantitative backscatter or even other techniques and nanoindentation, we have some real advantages. We're working at a similar resolution, and in fact, I'd argue that this is actually at sub-micron resolution, and my nanoindents are somewhere around the micron resolution. They're complementary, so I get really great compositional information, but I don't really get a lot of whys. I can also do 2D mapping. So in theory, I could look at this and say, okay, I know this is going to be stiffer in these wider regions. I'm not going to have any stiffness at all there, et cetera. But it's still not explaining why. The other problem is when I'm using quantitative backscatter and other techniques, it's technically challenging. Aligning my arrays, especially of a very large 2D array, there's a huge potential for error. One, a fraction of a degree of misalignment means that I've misaligned my whole array, and I'm getting the compositional information off of the wrong sites. So the other problem is it's really expensive, and unless you have an army of very talented and diligent graduate students, there's a potential for problems. So what I'm getting to is that we really have a need to combine these assessments into one single platform, and so that's what I've tried to do. So I looked at this problem a couple of years ago, and I said, well, why aren't we taking, Eve's study, for example, and just putting a Raman in conjunction with a nanoindenter, because that makes sense to me. And so let me talk just for a second about Raman spectroscopy and what it does. In the interest of time, I'll go through this quickly, but if you have questions later, you can ask. But it uses conventional objectives. You basically excite your sample with monochromatic light, or in other words, a laser of a specific wavelength, and you get something called a Raman shift. And so For some materials, there is no Raman shift. It turns out for all the materials we're talking about in this conference, especially the polymers and the tissues and things like that, you do get a Raman shift. And so what that does is you get this elastic scattered light that is deviated off of the surface, and you can measure that with some really high performance detectors and analyze it with your really nice fancy software that you get from your Raman company. But the point is that you get a spectra over certain wavelengths that you prescribe. And so what we're doing is we're basically in my lab using a near UV laser, and there's another shorter wavelength laser that near IR, I apologize, laser to collect spectra and then to interpret those. And I'll tell you how we're doing this in conjunction with our materials. So what is it suitable for? Just to review, we can study minerals. So our geology department just bought a really nice Raman system that's a little different than ours, so we're really excited to have 2 on campus. But you can look at polymers and ceramics, cells, protein, extracellular matrix molecules. I've seen papers where people have looked at nucleic acids using Raman spectroscopy. Really neat technique. The other nice thing about it is that it collects from a very small sample volume. So sometimes you can get down to less than a micron in diameter. The point is that it's pretty consistent with the nanoindenter spot size, essentially. And within that volume that you're sampling, you can identify species that are present. So unlike FTIR and other techniques, you don't really have to fix or section materials, and the water doesn't generally interfere with the Raman spectra. So in other words, if Michelle has a composite hydrogel material that she wants to perform Raman on, it doesn't have a beautiful surface, it's wet, she could put it in a Raman and get chemical information. It is a little bit of a problem because with imaging tissues, you have to use a longer wavelength laser because it's a laser, it heats up your sample, so you can get potential tissue damage. The other thing is that you get autofluorescence from collagen. So anyone who's ever imaged collagen in a confocal microscope, a tissue in a confocal microscope, the collagen signal is overwhelming. And so you have to use a longer wavelength laser, which means it's lower power, which means you have to sit on your sample for longer and things like that. So it's not ideal, but it's pretty good. It's good enough for us to want to do. The nice thing about it is that it penetrates into the sample, which is great because you get a sampling volume, kind of like nanoindentation. The problem is if you have one tissue sitting on top of another, that's of course confounding. It's also an issue in nanoindentation. And you have to have long collection times on the order of minutes sometimes. So that's sometimes good, sometimes bad. If you're trying to analyze a time-dependent phenomenon, that could be an issue. But the nice thing is the way that these systems are designed, you get a spot size that's consistent with nanoindentation. Okay, so what we did was I led a group to write an MRI proposal, or a major research instrumentation proposal, to acquire a Raman instrument and a nanoindenter. I wasn't sure it was going to work because with acquisition, you're supposed to buy an off-the-shelf component, and I said we were going to do some engineering. It did work. I ended up working with Heisatron, where we now have a TI 950, and we just have in crates that just arrived this Renishaw system. And I went to Heisatron about a week and a half ago to see them work together. So let me just show you a little bit about how they work. So they basically integrated 2 fiber optic probes into the optics on the nanoindenter. So in other words, they can maintain the noise floor and all the features that they really like about the nanoindenter, but these fiber optic probes and 3 USB cables are what connects to the Raman system. And so that means that they had to create a custom mount for the fiber optic probes. This is really heavy, so when we do dynamic testing, we have to actually take it off and put a normal objective setup on there. It's also got a 50x objective on here, so time will tell if we can ever find the sites that we want to indent. I think that we're going to have to switch them out. And then they also had to coordinate between the 2 companies to allow Heisatron and Renishaw to communicate stage coordinates so we could do 2D Raman mapping. So while I was out at Heisatron, we did a quick test study on ultra-high molecular weight polyethylene. I'll just show you a couple of results from this, and in the interest of time, you said I should go over like 5 minutes, right? Okay. For the record, I have permission. So ultra-high molecular weight polyethylene is the surface that George was showing earlier on some of these hip implants. In fact, it's favored over, people used to use Teflon. And it's because you can control the properties really well by controlling the processing conditions and the resulting crystallinity. The problem is where on these surfaces causes changes in the crystallinity, which also causes changes in the properties and the wear parameters of the materials. So so Yaroslav from Hysotron, he's one of their European engineers, was over here looking at ultra-high molecular weight polyethylene and looking at these crystalline domains, amorphous domains, and intermediate domains, and he was trying to understand what we could determine about modulus and Raman spectra of these in our new system. We were just trying to do some feasibility studies. So I'm going to skip this slide. If you're interested in reading a little more about the different phases later, I'm happy to share. So this is a typical Raman spectra from ultra-high molecular weight polyethylene. I pulled it out of this paper, and please note that the x-axis here, the wave number, is backwards because everything else I show you from now on will be forwards. I just didn't have the time to flip it around. But what they're showing is an unworn versus a worn in red polyethylene knee prosthesis. So you can see that the peaks shift a little bit in terms of their height, and maybe sometimes their placement. I don't think we actually have a shift in wave number on this plot. But this is like a lot of other spectroscopic methods where you'll look at peak height, peak width at half height, and parameters like that. There's a region, though, on here where they're really interested. So if you look at the full spectrum from almost zero to almost 3,200 or so wave numbers, you can see that there's a region here that's really most interesting. And this is the region that they were mapping. Note that my x-axis is now going in increasing order. And so I've labeled these peaks on this axis as to the ones that we want to see. And they've shifted around. I don't know why. Sorry about that. But the point is that we can look at these peaks, their locations, and learn something about the crystallinity and the chemistry of the materials that we're studying. And so here, I'm sorry?
The height of the peaks.
Oh, the height of the peaks, it's basically like a spectral count, how many times you hit at that wave number. So that's why you have to sit for longer periods of time to get your peak height to be higher and higher. Okay, so to be high enough to actually measure, in other words. It's a statistical sampling type of assessment, okay? And so in going and looking at specific peaks that are indicative of crystallinity, of crystalline regions, intermediate regions, and amorphous regions, what we can do now is we can correlate this to nanoindentation modulus. And this is just a conventional nanoindent Berkovich test. I mean, nothing special about it. Just to get this reduced modulus here that's shown in blue over 4 different samples of varying crystallinity. So when I looked at these slides that Yaroslav sent me last week, I thought, I have no idea what the difference is between these 4 materials until I started looking at this. So I think the take-home message can really be found in this orthorhombic, or the red plot, where we're actually decreasing in crystallinity and we see a decrease in the reduced modulus. And so that's really nice, because what this does is it shows promise that we can start going and collecting in this one system in situ information about the chemistry and the properties of the same material at the same time, putting the material on the nanoindenter stage without moving it. So I think in total, this study is nice. It shows feasibility of this technology, but it's not really as interesting as the work that we could do, because we have potential of looking at a region of a material, like a crystalline material, getting the Raman spectra, indenting it, changing the crystallinity potentially, looking at it again, and seeing different changes in structure and materials and things like that. I think that it's really exciting. And so some of the types of materials that I'd like to look at, this is a bone to ligament interface published by Helen Liu and Steve Thomopoulos, where they've basically done histology to show the true interface. This is FTIR, which is similar to Raman spectroscopy of that same interface. So you can see, well, of a similar region, where you can see the bone regions. The red are the marrow spaces, the blue is actually the material. Here's the fiber cartilage area, and this is where you get into the ligament. And then they're making these scaffold materials out of various types of mineralized material, hydrogel platform for a lot of the other components. And I think that there's great promise there, because again, to get these measurements where they've taken the native tissue, they've gone and done FTIR, they've had to take different samples, different slices. Adele works across New York City from Helen. I mean, this is a very complicated thing. And so now we have at least a little bit of potential to be doing this all in one place. And so there's a lot of potential for the different types of materials and the different questions that we can ask using this type of system. Okay, so I'm going to skip this next slide. So I guess with that, I'm going to finish up since I'm 6 minutes over. I think you gave me 5 or 10 or something. It was nebulous. And I want to just thank my funding sources, which is mostly NSF for this, and then all the people at CU Boulder and the departments and programs who help to contribute to the matching fund, because if you've ever put in an MRI proposal, you'll know that you have to have a substantial amount of matching funds for this type of thing. But I'm just really excited to come back and talk to all of you people in different conferences in the future with new data from this tool in the future. So I guess with that, any questions?
We have questions, yeah. Pick up something.